Of you. This video is about the processing of the tRNA, and in my previous video we have discussed the processing of the rRNA and the overall process of the RNA processing. So in this tRNA, genes of both prokaryotes and eukaryotes occur in clusters, and each cluster has specific gene for the several types of tRNA. So in case of E. coli, many of such genes cluster are arranged in operons, and genes have a cluster are transcribed into a single RNA transcript. And in mammalian, that is eukaryotes, the mitochondria, the tRNA sequence are used up as processing signals as they separate the sequence of a different structural genes. So tRNA precursor may be of two types, that is type 1 and the type 2 molecules. So in which type 1, the CCA sequence are at 3' terminus and are processed by RNase D to generate their 3' end. And the second one is the type 2 molecules don't have the CCA 3' terminal sequence, and type 2 precursors are processed at their 3' end by the RNase D or some other enzymes till the appropriate site is reached. The enzyme tRNA nucleotidal transferase is then added to the CCA triplet at 3' end for the further action. So it can be better understood through a figure. So in this figure, the introns are removed by the endonuclease enzyme and forms the two sub ends, that is 5' OH end and the 2' 3' phosphate end, which is a cyclic. So it can be separated by like this 3' OH end and the 2' phosphate end by phosphodiesterase enzyme, and then it ligates with the 5' OH end, and it forms the pre-tRNA to the mature tRNA form, and then 2 dash end for phosphate removes by phosphatase enzyme. And in this figure, the processing of pre-tRNA is done by the enzyme RNase P and 3' trailer end by the RNase D. There is a leader and the trailer sequence which is removed by the RNase P's and the RNase D and forms the mature tRNA. So all about the RNA processing, tRNA processing. Keep watching. Thank you.